Bonjour and welcome. I'm John Pereira, President-elect of the Canadian Pain Society, and it is my honor to interview Professor Jeffrey Mogil of McGill University. He is a world-renowned expert in pain genetics and the author of over 220 articles and book chapters. The Canadian Pain Society has just presented him with a Distinguished Career Award. Dr. Mogil, congratulations.
I'm very honored. CPS has been a very important organization for me for a long time. So this is a lovely honor, one I hoped and wished would happen one day.
What really struck you in your career as the first major breakthrough in terms of your research that you're really proud of?
Well, to be honest, I think it's probably the first or second experiment I ever did in graduate school. And it's not that the finding was so amazing, but the type of finding it was ended up being a theme throughout my entire career. So we were doing an experiment, and I was sort of helping out a postdoc in the lab who was in charge of this experiment. And we were working on a phenomenon known as stress-induced analgesia, and we were using a drug which was a very new drug at the time. It's a very famous drug now in science. It's called MK801. It's a NMDA receptor antagonist. And back in the day, NMDA receptor antagonists were new. We had some reason to believe that stress-induced analgesia might be mediated by NMDA receptors, and so we were using this drug. And the experiment was almost done. It looks like it was kind of working. And one late afternoon, early evening, I was killing time. I was meeting friends at a bar a little bit later, and I had time to kill. So I'm working on the computer and running stats on the data we had collected. And I decided to run a two-way Anova in addition to the one-way Anova that was called for by the experiment. And in the two-way Anova, I looked to see if the drug effect interacted with sex. And what I found was that on average, it looked like the drug was working okay. But what was really happening was the drug was absolutely completely working in male mice and absolutely completely not doing anything in female mice. And I remember getting very excited about that. So I had a highly significant two-way interaction. I remember the next morning, the postdoc came in and I told him what I had found. And I thought he'd be excited about it, but he was not excited because I had just taken a nice simple finding and made it complicated. And I'll never forget what he said to me. He said, and I'll try to do it in his accent, he was Polish. He said, "Jeff, sex differences are to enjoy, not to study." And he was kind of joking, but he was also kind of serious, and tried to convince me over the next hour or so that the sex difference wasn't even remotely as interesting as the drug effect. But I, of course, thought he was entirely wrong. I thought the sex difference was much more interesting than the drug effect, and followed it up. And, in my career, obviously, sex differences, of which that was the first one, has been a major theme. And sometimes we go looking for them, and sometimes, like that first experiment, we just find them by accident because we always use both male and female mice.
What you're saying is so critically important, given that the majority of people with chronic pain are women. How has your research affected how we think about these differences?
I think my research has sort of turned into advocacy there, in a way. So you're right, women are the clear majority of pain patients. This we've known for a long time. You also won't be surprised by the fact that the vast majority of preclinical pain research has historically been performed on male mice and male rats. I did a survey of that back in 2005, looking at the decade leading up to 2005, and I found that almost 80% of studies published in the journal Pain using rats or mice used only male rats or male mice. And then I did that again in 2015 and found that in 2015, again, almost 80% of papers published in Pain used only male rats or mice. I just published a new review on the topic. It came out online about a week ago. And we updated it and found that by 2019, the number had gone down from 80% to 50%. There's still too many studies that only use male animals, but there are less than there were before. So why has that happened? Well, I'd like to take credit for it, but mostly credit goes to governments. Canada changed its policy in 2006. The United States changed its policy in 2016. By 2019, things are starting to catch up to those policies. I'd like to think that at the very least, I've given governments pretty good ammunition to handle the backlash that they've experienced when they've tried to put in those policies. And so scientists don't like to be told what to do, and they don't like to change what they do. But when they argue that it's not necessary to study females because it doesn't really matter, governments can point to some of our studies and say, hey, wait a minute, it really does matter. And if you only use male animals, you're going to be missing stuff. It's going to be wasting the taxpayers' money. And frankly, I've believed for a long time is unethical.
This is absolutely fascinating. When you talk about the changes that need to occur in research, you mentioned one right now. What other changes would you recommend that we embrace?
It's interesting that you've asked that question in that way, because looking back on my career, I increasingly think of myself as not only a scientist, but sort of a meta-scientist, right? Trying to do experiments that shed light on how experiments should be done. And I think I've done it a few times in my career. The sex difference thing is one of them. Before that, I and my collaborators were among the first to show pain researchers that it mattered what strain of mouse they used. So you get different results if you use C57 Black 6 versus 129 versus CD1. All the different available strains give different results, and they can't be substituted for each other. I've done a lot of work in my career showing that environmental factors in the laboratory can matter. Things like injections causing stress that people didn't think of can affect findings. Whether animals are habituated or not before they're tested can affect findings. There's been a lot of findings that we've published where the point of the finding is to serve as an example that things that people were taking for granted and weren't paying much attention to can actually matter a lot in pain research, but also more generally in biomedical research.
Researchers often talk about how if they just had a little bit more time, there is one thing they just would love to study. For you, what is that one thing you would love to study in the world of pain that you just never got around to?
I think it's what I'm starting to do now. The reason I never did it before was because it's really expensive and it takes forever, and that's to look at the effect of pain on mortality itself. The epidemiologists are showing, and the data are more clear now, that pain decreases lifespan. And I think this is hugely important because one of the reasons that people use to say that pain isn't as important as other things like cancer is that they say that pain is annoying, but it doesn't kill you. But it turns out that pain does kill you, just doesn't do it directly. It does it indirectly. And again, as I said, the epidemiological data supporting this statement is getting stronger all the time. But no one has the slightest idea of how that might be working, what the mechanism of lifespan reduction is. And so we've started with what we hope is appropriate statistical power to have mice live their whole lives in chronic pain and see how long they live, and then see what they die of, and try to work out the mechanism of how pain leads to mortality. And again, I've been interested in that idea ever since I was in graduate school, and John Liebeskin, my advisor, published an editorial in Pain called Pain Can Kill. But I've never been able to do it because to do that experiment requires that you pay for the upkeep of mice for 3 years until they die and hope that you have enough sample size. It's a very expensive and, very long-term experiment. It's not one that you could give to a grad student or a postdoc, for example. But now that I've gotten to a point in my career where I'm, patient and willing to wait, we've actually started in earnest trying to figure this out.
Is it possible that proper pain management can save lives and make people live longer?
Yes, absolutely. And of course, that's one arm of the experiments. It reminds me a little bit of the finding that got a lot of attention a few years ago when it started to come out that chronic pain was decreasing gray matter in the brain in chronic pain sufferers. And this was thought to be important again because this was something objective and tangible that people could point to that is a problem with chronic pain other than just the subjective morbidity of it. It could be measured. It was perfectly objective. Everyone assumed, I assume rightly, that gray matter decreases are bad. And why I'm telling this story is that a paper came along done here at McGill by Laura Stone showing that, well, if you treat the pain, the gray matter comes back. It's reversible. And so I wonder if that might not also be the case for pain's effect on mortality.
When it comes to the effect of pain on gray matter, do you see this as a reason for a more holistic approach to pain?
I have complex feelings about it's not so much the biopsychosocial model. Obviously, I totally believe in the biopsychosocial model. I just worry that when people say they don't like reductionism and they want to replace it with quote unquote holistic approaches, I don't really know what those are. And so I think that we need people to move forward on all fronts. It is true that treatments aimed at higher level functions and affect and social factors can effectively treat pain. They don't do it very well. The numbers needed to treat there are even worse than the drugs. Drugs, of course, don't have great numbers needed to treat either, and they also have side effects. I haven't given up on the concept of a magic bullet, to be honest. I know that's an unpopular statement in 2020, but it strikes me that although nothing has been even close to a magic bullet so far, there is absolutely nothing that rules out the idea that some molecule will be found that when blocked or activated will have widespread, highly effective, effects on pain. The fact that we haven't found it yet may only be due to the fact that we've only investigated a tiny, tiny fraction of the molecules that genetic approaches have now shown are involved in pain. So I think that, the molecular folks should plow ahead looking for molecular treatments, and the behavioral folks should plow ahead trying to refine and make the behavioral treatments more effective. And I wish that we didn't think of these as being opposed to each other, but rather different people trying different things. And, history will be the judge on what ends up being most successful.
You believe that a magic bullet is possible for pain, and to the researchers listening to this podcast, where does your intuition tell you they should start to look?
I stopped picking winners a long time ago, but I do believe that the universe of potential targets is much, much bigger than we thought. I'll tell you a little story. So when genome-wide association studies were new, and only 3 or 4 had ever been done for pain, I was giving talks periodically on genetics around that time, And what I used to do was put up the names of all the genes that people had found to be potentially involved by that approach at that time. And so I made a slide where the screen was filled with the names of genes. And then what I did was I animated it so that I pressed a button and the gene turned green if we knew about this gene, right? If you did a PubMed search on the protein and it came up with 3 or more hits, that gene would go green. If the PubMed search turned up no hits at all, so it was completely novel in the field of pain, the gene would go red, and then in between the gene would go yellow. And I did this for effect because the whole screen turned red. Basically, all the genes that had come up in GWASs were genes that we've never heard of before in pain. And I said to the audience at that point in my talk, this means one of 2 conclusions is true. Either GWASs don't work, either the technique is just giving us garbage as results, or all these genes really are involved in pain. And the fact that we've never heard of them means that we only know a small fraction of the total number of genes that are involved in pain. That is to say, we only know the tip of the iceberg, and there's a big, huge iceberg underneath. At the time I gave that talk, I actually believed that the right answer was that the technique was giving us garbage, and that GWASs should be ignored. But I think I've changed my mind, because in the interim, 2 separate times, grad students have come up to me and said, I was at that talk and I'm actually working on one of those genes that no one had ever heard of. And I made some sort of knockout mouse, or I knocked it down with a virus, or however they manipulated these genes. And they would say to me, and the resultant mouse is crazy insensitive or more sensitive to pain. That is to say, they were highly successful in proving that that gene really is involved in pain. And so now I believe that the 50 proteins that we think are involved in pain, that we've all been studying for decades, are 50 of maybe 4,000 that are actually involved. And that means that there are almost 4,000 more targets. Any one of them might be a magic bullet. Most of them probably won't, of course, but there's a lot more chances for us to get really, really lucky.
With all your experience in the field, where do you see us being in terms of pain research 20 years from now, in the year 2040?
Well, we've got 2 problems in pain research. One is that pain is in the brain, right? And that means it's neuroscience. And you know the old joke about neuroscience, right? Neuroscience is the science of the future and always will be. So there's some reason to believe that anything mediated by the brain is probably too complicated for us ever to get a decent handle on. And that, of course, would include pain. Pain is doubly handicapped by the fact that, first of all, it's fundamentally subjective. I'm not sure we're ever going to get a truly objective biomarker of it. We're handicapped by the fact that it's being processed in areas that are inaccessible in humans. You know, if you study cancer, a lot of the relevant biology is going on in the tumor cell, which the cancer patient is more than happy for you to take and study. Pain patients presumably are a lot less enamored with the idea of taking biopsies of their dorsal root ganglion or their spinal cord or their amygdala or their anterior cingulate. So we have a lot of, handicaps trying to get to the same place as other branches of biomedicine. 2040, it's 20 years away. I'm not sure anyone's going to find my magic bullet in the next 20 years. I think we're going to make slow and constant refinements, and we're going to be able to get the numbers needed to treat down for both our pharmacological therapies and our behavioral therapies. Now, if you ask me about 2080 or 2100, by then I would be a lot more optimistic that if there's a magic bullet to be found, we'll have found it.
And finally, let's end with a question for those listening that are thinking of following in your footsteps, going into pain research. For the trainees, what advice would you give them?
It's kind of tough because everyone that has become successful has become successful, I'm convinced, mostly due to luck, not because we were any smarter than anyone else, but we happened to be in the right place at the right time. And at the very least, we had enough competence not to mess it up when the chance was given to us. The way I got lucky was I happened to be the first person who was ever trained in both pain and genetics. In graduate school, I got pain training and got interested in genetics, and then I did a postdoc with an actual geneticist. So that's great. So I was able to put two things together that had never been put together. And for a while, no one cared, but then suddenly they did. When transgenic knockouts were invented and people started to note that the results that you would get from testing those mice would depend on the genetic background of the mice, people started to say, okay, what do we know about how different strains react to pain? And I turned out to be the only one who had ever collected that data. And suddenly I had a career. I guess I still would recommend that students try to find some intersection that has never been occupied before and situate yourself at that niche and then hope that that niche becomes important. I think all the, dual niches are probably gone by now, right? People might have to find the intersection of three things in 2020. So it gets ever harder. And I guess the corollary of that is that people shouldn't beat themselves up. If it's not going well, that doesn't mean you're not good. It just means you haven't gotten lucky yet. Are you going to get lucky one day? Well, I don't know, but you might. And if you love science and you're willing to wait around for your shot, it certainly is an amazing profession and an amazing lifestyle.
Dr. Jeff Mogil, you are the Canadian Pain Society 2020 Distinguished Career Award winner. Thank you for joining us. And once again, congratulations.
Well, thank you very much. And I really want to thank the Canadian Pain Society for being a very important part of my career for the past 20 years.
You can learn more about the Canadian Pain Society, our vision and mission at canadianpainsociety.ca, Twitter @CanadianPain.